Thank you very much. I actually did not come alone this morning. I brought with me my doppelganger, Dr. Gero. He's also a scientist, a mad one in this Japanese comic called Dragon Balls. He strives for world domination, just like I do. And if you look carefully, you can see that his skull has been replaced with a transparent plexiglass dome, so that the workings of his brain are open to optical control. And that's, of course, what this is all about. We modify specific nerve cells genetically so that their function can be controlled with light. And these two essential ingredients, genes and photons, have given the method its name, optogenetics. Before I explain how it works, let's just think for one moment why being able to control the brain is important. If you like puzzles, and as a neuroscientist trying to understand how the brain works, you'd better, you probably know that staring long and hard at a difficult problem rarely gives you the answer. It's often much more productive to play with the problem, to try different solutions, to see whether you can find a pattern that makes everything fall into place. But I think it's fair to say that historically, neuroscientists have mostly stared at the brain and not played with it. And part of the reason for that, of course, is the enormous complexity of the nervous system. Even the brains of the fruit flies that we study in my lab are built from a few 100,000 nerve cells of many different flavors, with millions of connections between them. So where to begin? But there's one organizing principle that's behind this seemingly impenetrable complexity, and that is the genetic blueprints that orchestrate the development of the brain. And the key idea of optogenetics is to use this blueprint as our platform for our experimental manipulations. We use specific genetic markers to single out particular classes of nerve cells in the nervous system, and then use them to place into these neurons ion channels that are just like the ion channels that are responsible for all the electrical signaling in our nervous systems, with one important exception. They are coupled to light receptors, so that when a photon of light strikes the receptor changes shape. That shape change is transmitted to the pore of the channel opens, a small current flows, and the neuron fires an electrical impulse. Now, what motivated the invention of optogenetic control was the idea that a technology like this would open 3 experimental doors that had previously been locked. The first is the identification of the neuronal causes of behavior. If you could play back particular electrical signals into the nervous system and recreate perceptions, movements, emotions, memories, thoughts, then you could credibly claim that you have understood the neural signals that normally underlie these aspects of our mental lives. The second locked door is the search for neuronal connections, which is key to deciphering the wiring diagram of the nervous system. By scanning a light beam across neural tissue, this can be done much more efficiently than in painstaking searches with electrodes. And finally, there's the possibility of testing mechanistic hypotheses. If you had an idea how something worked, then of course the way you find out whether you're right or wrong is through directed interference with the system. So in order to illustrate what's now possible, I've chosen one aspect of our recent work that I think shows how optogenetics has allowed us to open all 3 of these doors. And that problem is the control of sleep. Sleep is one of the great biological mysteries. Each night we disconnect ourselves from the world for 7 or 8 hours, a state that leaves us vulnerable and unable to do what we need to do in order to propagate the species. But despite these risks and costs, we still don't know what sleep is good for. And I think the way to go about finding out what that vital function of sleep is to look for the mechanisms in the brain that normally regulate sleep and waking. And there's 2 of these mechanisms that are symbolized on this diagram by 2 different forms of oscillation. The sine wave symbolizes the circadian clock, which oscillates in synchrony with predictable changes in the outside world that are caused by the Earth's rotation. As such, the clock is an adaptive mechanism that makes sure we do our sleeping when it hurts us least, but it can speak to the deeper mystery of why we need to sleep in the first place. The answer to that mystery will come from understanding the second controller, the sleep homeostat, which is symbolized here by the sawtooth oscillation that's superimposed on the clock. While we are awake, something, and we don't know what that something is, happens in our brains or our bodies. And when that something hits a certain ceiling, we go to sleep. The system is reset during sleep, and then the cycle begins anew when we wake up. Now, we know a lot about the circadian clock, and this is really the Rosetta Stone that broke the problem open. The discovery by Seymour Benzer and his graduate student Ron Konopka of fruit flies whose clocks ran abnormally fast or slow. And from that discovery really came, through the work of many, many laboratories over the past 45 years, a pretty complete molecular, cellular, and systems understanding of circadian timekeeping. This slide, in contrast, summarizes pretty much all we know about the sleep homeostat. So my goal for the rest of this talk will be to try and draw at least some outlines on this blank canvas. The story begins with the discovery by a former postdoc in the lab, who is now a professor at the University of California in Los Angeles, of sleep control neurons in the brains of flies that exert a powerful influence over sleep and waking. Optogenetics has taught us that these neurons represent the output arm of the sleep homeostat. The way we do these experiments is we take a fly and we head fix it to a mount and then let it walk on a little treadmill, a styrofoam ball whose rotations we read out with an optical computer mouse. Now, since there are no documented cases of somnambulism in flies, we know that when the fly is walking, it must be awake. What you can see is that the head capsule is open, that we've inserted a recording electrode into one of these sleep control cells and expressed our light-gated ion channels in the entire population, all 2,000 or so of them. And here's an experiment lasting for half an hour. You see that during the first few minutes, the sleep control neurons are electrically silenced. They don't emit any impulses, and the fly is walking alone happily. But as soon as we switch on the lights and the sleep control cells become electrically active, all movement stops, the fly goes to sleep. When we switch the cells off again, the fly wakes up and starts to move, switch the cells back on, and the fly goes back to sleep. So we've isolated a switch in the brain that allows us to toggle the animal into and out of sleep on command. Now, in further studies, we discovered that these sleep control neurons normally exist in two states. One state in which they're electrically active, shown here on the left, and another state in which they're silent. So this then, of course, led us to the idea that perhaps this is the way how homeostatic sleep control works, by switching these neurons between these two states. And in fact, if we sample many different animals and check in which state the sleep control neurons are in, we see that in sleeping flies, the sleep control cells, as expected, tend to be electrically active, whereas in awake flies, they tend to be quiet. But in order to really substantiate this point, what one would like to do is switch these cells deliberately from one state to the other, isolate the signals that normally drive the switch, and understand how it works. So what we need is a signal that allows us to control wakefulness and the state of these neurons. Now, a first clue of what such a signal might be had come many, many years ago in the first experiments that showed optogenetic control of animal behavior. These experiments were done by my then graduate student, Susanna Lima, in 2004. Susanna had expressed our light-gated channels in all neurons in the brains of flies that communicate through the molecule dopamine, and then measured the activity of the animal. What you see plotted here is the movement tracks of 4 flies, each column is 1 fly, before and after switching on the dopamine system. And you see that the flies become highly aroused as soon as the dopamine system is activated. That's consistent with the actions of dopamine in our own brains. All drugs that act as psychostimulants, such as amphetamine or cocaine, do so by elevating dopamine levels in our brains. So if that's true, if dopamine is the arousal-promoting substance, then of course you would expect that it switches the sleep-inducing cells off. And there's in fact a dopaminergic neuron that projects into exactly the same region that's inhabited by the sleep control neurons in the fly's brain. In fact, the two cell types shadow each other so tightly that the question naturally arises, might they be connected? And the way we probe for connection is, of course, by recording the activity of a sleep control neuron while we optogenetically stimulate the dopaminergic neurons. And here's what we found. This is a sleep control cell in a sleeping fly. The cell is electrically active. Then we switch on the lights, drive dopamine release, and you see that the sleep control neuron switches to the electrically inactive state and the fly starts moving, it wakes up. If we wait a little while, the animal often spontaneously goes back to sleep, and that transition back to sleep coincides with the return to the electrically active state of the sleep-inducing cell. Now, what about the mechanism that underpins all of this? I'll just summarize a lot of work in the form of a simple cartoon. What we have found is that sleep control neurons in the electrically active on state contain a molecule we've discovered and dubbed Sandman. And what dopamine does...
A candy-colored clown they call the Sandman tiptoes to my room every night just to sprinkle stardust and to whisper, go to sleep, everything is all right.
So you see that Sandman in the electrically active state is sequestered inside these sleep-promoting neurons. When dopamine binds to its receptor, it causes Sandman to move to the outside of the cells. And there it acts like a short circuit that switches the electrical activity of these neurons off. That's it. That's the transition that drives the switch from sleep to waking. Now, before I conclude, I'd like to emphasize that what I've described is actually a device that's intensely familiar to you because you all have it on the walls of our living rooms. But this device does not measure temperature and switch on the heat when it's too cold. Instead, it measures something, and we don't know what that something is, and puts us to sleep when that something exceeds a certain threshold. The billion-dollar question in all of this, of course, is, what does the sleep homeostat measure? If we knew the answer, I think we would have solved the mystery of sleep. Before I hand over to the next speaker, let me just take you back for 1 minute into the very stone age of optogenetics. As we started to work on these remote control technologies, I discovered that I was not the only scientist who had seen the need for a method like this. The other one was Francis Crick, one of the two discoverers of the double helical structure of DNA, who in a paper that was published in the millennial issue of the Philosophical Transactions of the Royal Society, wrote, the next requirement for understanding the brain is to be able to turn the firing of one or more types of neuron on or off in a rapid manner in the behaving animal. The ideal signal would be light. This seems rather far-fetched, but it is conceivable that molecular biologists could engineer a particular cell type to be sensitive to light in this way. So when we had the first experiments that turned this far-fetched possibility into a reality, and these are these experiments, that's a neuron from the brain of a rat grown in culture, and it expresses a light receptor, and you see that when we record its electrical activity, it sits quietly in the dark, but as soon as we switch on the light, it responds with a volley of electrical impulses. So when we had these experiments, I sent a preprint of the paper to Crick, who replied in a style that apparently was his characteristic. If you read this wonderful book called The Eighth Day of Creation, which recounts the early history of molecular biology, you know that Crick was a prolific letter writer who steered the development of the field through a vast correspondence that combined two stylistic hallmarks, encouragement and constructive criticism. And that's exactly what I got. Crick wrote that he read the paper that I had sent him with great interest and was excited to see that the system already worked, at least to some extent. However, he realized, as I did, that it still needed improvement and that would take further work. Unfortunately, Crick did not live to see how these experiments progressed, but the improvements have come, not just thanks to our work, but thanks to those of many others who joined the field. But I think as a result, the way we neuroscientists go about our business has fundamentally changed. We have transformed the field from an observational into an interventionist discipline. Thank you.